Welcome back to Chapter 3. In this lecture we will cover the rest of the chapter. Last lecture we covered nucleic acids and proteins. In this PowerPoint we will focus on enzymes and how they participate in chemical reactions. A catalyst is a substance that speeds up reactions. Catalysts are not permanently altered. A catalyst does not make a reaction occur that would not otherwise occur. This means that a catalyst does not magically make a reaction occur. Given the proper molecules, these reactions would still occur without the catalyst. However, it is with the help of the catalyst that makes the reaction occur faster. Most catalysts are proteins. They're also more commonly referred to as enzymes. There are a few RNA molecules that work as catalysts as well. These are called ribozymes. Next, we're going to talk about chemical reactions. An exergonic reaction is a reaction that releases free energy. Without the catalyst, the reaction is slower because of the energy barrier between reactants and products. An input of energy initiates the reaction, which is called the activation energy, sometimes abbreviated as EA. This input of energy puts the reaction into what is known as a transitional state. This graph illustrates a change of energy in reaction. On the y-axis, you have the amount of free energy that is available, and on the x-axis, you have the time the reaction takes place. The reactant is stable by itself. With an input of energy to cross the energy barrier, also known as the activation energy, energy will become released from the reactant, thus decreasing the amount of free energy. Activation energy can come from heat to start the reaction. Hopefully, you all see a problem with this within a biological system. Heat would be very destructive and all actant reactions would be accelerated. Also, you may catch on fire, which is bad in itself. So organisms need a way around this. To get around this hot dilemma of being able to activate reactions, organisms use enzymes. Enzymes lower the activation energy by enabling reactants to come together quicker. For example, a molecule of sucrose in solution may hydrolyze into glucose and fructose in about 15 days. By adding the enzyme sucrase, the same reaction would occur in 1 second, which, as you can see, is a lot faster. This graph illustrates a reaction again, with the amount of free energy on the y-axis and the time course of reaction on the x-axis. But notice, with an enzyme present, the red line, the activation energy is lower than it is without the enzyme. This illustrates that enzymes lower the activation energy, thus helping the reaction move along quicker. Enzymes work on substrates. Enzymes are specific for only one chemical reaction. As you can see on the top picture of the slide, the enzyme is specific to one specific substrate. The substrate will bind to the enzyme as was known as the activation site or active site. The particular 3D shape of the enzyme and its chemical properties make it specific for only a certain substrate. Here in this diagram we have the enzyme sucrase. Sucrose is the substrate. The sucrase binds to the sucrose at the active site. At the end of the reaction, the sucrose is broken into the products, glucose and fructose. The enzyme sucrase is unchanged and ready to participate in another reaction if needed. Together, the substrate and enzyme make up what is known as the enzyme-substrate complex, also abbreviated as ES. This complex is held together by hydrogen bonds, electrical attractions, or temporary covalent bonding. You can see from the equation that E is the enzyme and S is the substrate. When they bind together, they make ES, or the enzyme-substrate complex. At the end of the reaction, the enzyme is unchanged, and a product P is produced. Enzymes may use one or more mechanisms to catalyze a reaction. One method is called inducing strain. In this method, bonds in the substrate are stretched, putting it into an unstable transition state. You can see in our picture here our purple enzyme and the olive green substrate. When they bind together, the enzyme will change shape, thus putting the strain on the substrate. An enzyme structure is so specific that it will only bind to one or a few substrates. This is due to the primary structure and how the amino acids interact with one another, as we saw in our last lecture. Enzymes change shape when it binds to a substrate. Many scientists use the analogy of a baseball catcher's mitt. When the enzyme catches the enzyme, it will bind around it, much like a mitt does when it catches a baseball. And at the end of the reaction, it will release it. Here is a complex model of an enzyme, with no substrate on the left. After it receives the substrate, the enzyme will change shape by bending around it. The change in enzyme shape occurs when its substrate binds at the active site. Some enzymes require ions or other molecules called cofactors in order to function. These cofactors may bind to the active site or another secondary site to turn the enzyme on. Some examples are metal ions, coenzymes, which aren't enzyme specific and can participate in reactions with many different enzymes, and prosthetic groups, which permanently bind to the enzyme. Table 3.3 from the book gives some specific examples of cofactors that are metal ions, coenzymes, and prosthetic groups. If you want a reaction to go faster, just add more enzymes that are specific for the reaction. However, there is a saturation limit where the amount of product from the reaction does not increase. This occurs at what is called a saturation limit. This is when all enzymes are filled with substrate. Increasing the amount of enzyme past this point will not make the reaction go any faster. To summarize what we have went over with enzymes will decrease the activation rate for a chemical reaction to occur. They do this by binding to the substrate. Over time, the reaction rate will level out because all the enzymes are bound to substrate. Alright, first progress check. Please take your time to answer these questions and continue on with the lecture. An enzyme-catalyzed reaction is usually just one step in a multi-step reaction. These reactions occur in metabolic pathways. The product of one reaction is a substrate for the next reaction, and each step is catalyzed by a specific enzyme. You can see one particular metabolic pathway above, the Krebs cycle. Each intermediate product is catalyzed by a particular enzyme. Cells have hundreds of enzymes that participate in interconnecting metabolic pathways, which forms a metabolic system. Metabolic pathways allow cells to regulate metabolism by controlling the amount of a particular enzyme. Cells have abilities to turn the synthesis of enzymes on or off. The diagram above is an example of a metabolic pathway. Starting on the left side, you have a simple molecule. Then you have enzyme 1, which produces intermediate 1, which goes on to the second reaction with enzyme 2, and so on, until you have a complex molecule at the end of this particular reaction. Our bodies have ways to regulate enzymes. It would be unwise to produce an enzyme that you do not need at the moment. There are also chemical inhibitors that bind to enzymes and can slow down reaction rates. Our bodies have natural inhibitors that regulate metabolism. Artificial inhibitors are used to treat diseases, kill pests, and study enzyme function. One type of inhibition is called irreversible inhibition. This is an inhibition that covalently binds to an active site. Remember, most substrates use ionic or hydrogen bonds when binding to an active site. Those bonds are relatively weak and can be removed so that the enzyme may function again. Covalent bonds are strong and usually not able to be broken apart. So, irreversible inhibition will permanently inactivate an enzyme due to it covalently binding with it. Some insecticides and poisons work this way. Here we have the insecticide EIPF, which permanently binding to an enzyme's active site. By binding permanently to the active site is effectively taking this enzyme out of commission. There's also what is called reversible inhibition. This is an inhibition that can be reversed. There are two types of reversible inhibition. A competitive inhibitor binds at the active site, but no reaction occurs. It competes with a natural substrate at the active site. A non-competitive inhibitor binds at a site that is different than the active site. One such site is called the allosteric site. This will cause a change in the shape of the enzyme. Figure 3.19 shows both types of reversible inhibition. On the left is competitive inhibition, where the inhibitor binds at the active site. This does not allow room for the substrate to attach. The right side shows non-competitive inhibition. You can see the inhibitor binding on the enzyme side, which changes the enzyme shape and does not allow the substrate to bind. Allosteric regulation is a form of non-competitive inhibition. The inhibitor binds at the allosteric site on the enzyme, which changes the enzyme shape, thus not allowing the enzyme to bind the substrate. Allosteric enzymes can regulate the activation or inactivation of enzymes. Here we have two examples of allosteric regulation activating an enzyme. On the left you have the activator binding to the enzyme's allosteric site, thus turning on the enzyme. On the right you have the enzyme protein kinase binding to an enzyme to do the same thing. Both these examples turn on the enzyme so it can bind to a substrate. Metabolic pathways, as we mentioned before, can get very complicated with many steps from the beginning product to the final product. We will look at two metabolic pathways, photosynthesis and cellular respiration, in more detail in future lectures. But know that the first reaction in a pathway is what's called the commitment step. When this occurs, all other reactions will happen in sequence, till it reaches the last reaction. The final product of a metabolic pathway usually acts as an inhibitor for the first enzyme in the sequence, which will effectively shut down the pathway. This is known as feedback inhibition. You can see in the general pathway pictured above you have 3 different reactions, each with a unique enzyme. The final product of the last reaction will inhibit the enzyme in the first one. This is one way our bodies can regulate their metabolic pathways. Here's another example of feedback inhibition. The starting product is the amino acid threonine, which is transformed into an intermediate product by an enzyme. The final product is the amino acid isoleucine, which will in turn act as an inhibitor for the first enzyme. This will effectively shut down the metabolic pathway. There are some environmental factors that can influence enzyme activity. One factor is pH. All enzymes have optimal pH at where they work at. Too high or too low can inactivate the enzyme. The concentration of hydrogen ions in the environment, which pH measures, can affect the tertiary structure of an enzyme. Temperature also influences enzyme activity. Increasing temperature can increase the rate of a chemical reaction, but if too high, temperature can inactivate the enzymes by denaturing them. Here we have 2 graphs. The top one shows optimal activity of enzymes in their pH. The reaction rate for the enzyme pepsin, which is found in our digestive system, has a higher reaction rate at a lower pH, while the enzyme chymotrypsin works better at a pH of 7 to 8. The bottom graph shows an optimal rate of enzyme and temperature. By increasing the temperature, the rate will increase, but increasing too much, denature the enzyme, thus turning it off. Isoenzymes catalyze the same reaction, but have different physical properties and compositions. Isoenzymes have different optimal temperatures or pH, which allow an organism to adapt to change in the environment. Rainbow trout, as an example, have isoenzymes that allow them to live in both warm and cold water, so that they may survive and have their metabolic pathways continue unhindered. Alright, here's our final progress check. Please take a minute or 2 to answer these questions. Okay, if you remember at the start of Chapter 3 in our last lecture, we opened up with the question, how does an understanding of proteins and enzymes help to explain how aspirin works? Well, we may not realize it, but we have enough information to answer this very question. Aspirin binds to and inhibits an enzyme called cycloogenases. Why is this important, you may be asking yourself? Well, it just so happens that cycloogenases catalyze the first step in the metabolic pathways that produces prostaglandins, which cause inflammation and pain, and thromboxans, which stimulate blood clotting and constriction of blood vessels. And as you know, pain is bad. So, aspirin binds at the active site of cycloogenase. While it binds at the active site, it changes the shape of cycloogenases, putting it out of commission, which reduces inflammation and pain. Here on the left, we have a picture of aspirin. The middle picture is showing aspirin binding to the active site cycloogenase. When it binds to that active site, it's effectively taking this enzyme out of commission, reducing pain and inflammation in the process. So with that out of the way, I'd like to end on a joke. What do you call an acid with attitude? Amino acid. Don't laugh all at once. Thank you for listening, and hope you have a great rest of your day.